You can start now, Sonia, if you want.
Pardon? Can I start it?
Yes.
It's correct. Well done.
Yeah. What'd you get?
Opa.
Oh. Go. The usual.
Why are you hitting her? Why?
She makes me go mental.
You didn't even say anything.
Alaida, maybe it's easier if everybody switch off their microphone.
Yes, I hope I managed to mute now. Yes.
Okay. Good afternoon, everybody. Or good morning. I don't know what time is in Mexico right now.
9 o'clock.
9 o'clock in the morning. Well, good morning and afternoon then. Well, let's kick our seminar, bilateral seminars start. My name is Sonia Correa Miller. I'm professor of neuroscience at Manchester Metropolitan University, and I am head of the neuroscience team.
We can adjust that for more than 120.
Research within the Bioscience Center, Research Center at the MMU. And so I won't have time to talk about my research today, but I'm very pleased to introduce two scientists, two neuroscientists, to talk to the seminars. And we hope that we will be able to follow up some of the research developed between our centers and establish potential collaboration. So first of all, also I would like to thank Araida for organizing these exciting seminars and introduction between our groups, and hopefully we can take that forward and repeat in the future. So for the talks this afternoon, so we have two talks, and hopefully we can hear a bit more about the research in both centers. So from the MMU, we're going to have Dr. Chris Morgentorich. So he obtained his PhD from the Ludwig Maximilian University from Munich in Germany, and studied for 5 years a postdoc at the Max Planck Institute of Psychiatry before joining MMU as a lecturer in 2012. So his research interest is focused on understand the mechanism by which environment exposure impacts on stress, neuroendocrine and neuroinflammatory pathways. And to do that, he combines epigenetics, genetics, in vitro and behavioral studies in animal models, and translating these findings, and his main aim probably is translating these findings to inform studies in humans. So, so I will then, no further ado, ask Chris for his talk.
Thank you. Can everyone hear me? Is this working?
Yeah, it's working fine.
Brilliant. Okay, so yeah, so my talk really is going to focus on the idea about animal and human studies. And one point I'd like to bring across in my talk really is the importance of animal studies in allowing to develop human hypotheses, and also the importance of human studies in allowing the development of animals. So it's this translational approach between the studies. They both need each other, and they both have their important uses. So hopefully some of my research can show that. So I'll go over a bit of the background. I don't know how much you know about some of this, but the HPA Axis and epigenetics, and I'll go through maternal separation mouse study, and then the maternal depression rat, and then the human work I've done from that, the pre and postnatal interaction between the human and the rat, intergenerational transmission and the rat studies, and some of the human stuff I'm doing at the moment, immune studies and social stress in rat and human, and some of my work on dementia, particularly in the gene-environment interactions, which I'll come to at the end. And hopefully, if I've got enough time, G by E animal models that I'm working on at the moment. But let's see how much time I have. So the HPA axis is the important axis, the your endocrine axis that regulates stress. So what you have within this axis is you have the hypothalamus in the brain, and this from the pituitary releases vasopressin and COH that act at the adrenal gland to release ACTH that stimulates stimulates cortisol production. So the cortisol production then regulates various parts of the brain and importantly provides a negative feedback mechanism through the glucocorticoid receptors to shut off the HPA axis. Now the HPA axis is extremely important. It's extremely important for protecting us against stress. The things that come out your bath plug sometimes, which scared the bejeepers out of me, no end. But the important thing is you have the stress reaction, and this is a good stress, and it's not something to be ashamed about. Well, it's a bit embarrassing. My wife has to get rid of it for me, but it's not something you're ashamed about. Your stress system works, and that's what it's there for. Now, this is the bad stress. Papers will not fall on top of you and kill you. This is the bad stress. This is the chronic stress. This is the stuff we really got to manage. And so the idea about this is when you get that kick from the HPA axis system, your stress kicks in, you're not able to turn this off. And that leads to the chronic stress we often associate with so many mental health disorders. So the idea we're working towards really is this idea about adaptation. So the idea is that we need to adapt our HPA axis to adapt to levels of stress we perceive that we would be subjected to later in life. So during prenatal and postnatal life, we adapt our HPA axis. If we're expecting high levels of stress later in life, because we have high levels of stress earlier in life. So what we do is during particular periods of development, we have this adaptation where stress interacts at the genome to program through the epigenome particular endocrine circuits. And these endocrine circuits give us an neuroendocrine phenotype. And this could be a very good adaptive system. We're adapted to our later environment. This could also be a very bad and maladapted system as well. So for example, you've got a high level of stress early in life, but we don't have that level of stress later on in life. So constantly we have a HPA axis, which is a hyperactive, and this leads to susceptibility to mood disorders. So the idea when I first started a lot of this research was that early life stress could epigenetically program the expression of key genes regulating stress. So epigenetics, in case some of you that may be quite new to this, is an important aspect of the genome. All genes are epigenetically regulated in all tissues. It's not just one gene here and one gene there. All of them are. So within all of our genes, with all our tissues and all of our cells, we all have the same genes, obviously. So your toenail to your neurons have exactly the same genome. But only about 10 to 20% of those genes are active in any cell. You've got neurotransmitters that are active in the neurons, but not active in the toenail. And even though the gene is there, it's not turned on. And this is epigenetics. So you may get a gene for alcohol dehydrogenase, which is in every tissue, but it's only expressed in a few tissues. And that's what epigenetics does. So epigenetics describes DNA methylation and describes histone modification. There's also microRNAs and a few other things as well. But these are the main sort of mediators of epigenetics. There's a number of factors involved in epigenetics. So usually it happens at a CG residue. This is the CG sequence. You've got enzymes which methylate those regions. You've got enzymes which demethylate those regions. When these regions are demethylated, transcription factors can bind and turn a gene on. When they're methylated, transcription factors may no longer bind, and you may get other proteins, methylation binding domain proteins or chromatin remodeling proteins that will turn a gene off. So what I've been looking at is really human studies and rodent studies and translating these studies and findings backwards and forwards to push really research in understanding how early life stress and early life changes program long-term outputs, particularly in the regulation of stress. So one of the first studies I looked at was maternal separation in a mouse. So separated the mother from its offspring for about 3 hours each day for the first 10 days of life. Now, following that, looking at 6 weeks, 3 months, and 1 year, I can see that vasopressin gene was constantly upregulated in those animals separated from the mother. ACTH was produced higher, glucocorticoid was increased, and you can see high levels of corticosteroid. So what you can see is even 1 year mouse separated from the mother for only 3 hours a day, so that they received enough nutrition, everything else was okay, they just didn't have the mother's care for the first 3 hours, the first 10 days, had a permanent level of HPA axis activity. So a permanent level of stress. By looking at methylation, I saw in the early life stress mice at 6 weeks old, they had less methylation. They had more vasopressin expression, less methylation at the promoter, if you can see here in the corner. This methylation was similar during the 10 days, and I was able to show that a protein called methylation binding domain protein does not bind in the early life stress mice to the vasopressin promoter. It binds in the control and it recruits methylation. In the early life stress, it is phosphorylated by commodulin kinase 2 in the PVN, particularly the neurons expressing vasopressin, through depolarization. So what happens is early life and stress, the neurons in the PVN are activated. This activates commodulin kinase 2. This inactivates MECP2. MECP2 can't bind to vasopressin. MECP2 can't shut vasopressin off. Vasopressin remains high, and you get high levels of stress in these mice throughout later life. Developing to try and understand further how MECP2 is recruited, I developed an embryonic stem cell system to produce hypothalamic-like vasopressin neurons. And it also used very early prenatal pups as well to understand what comes before the MECP2. So combining these together, I'm able to develop this pathway where what we have is in prenatal development, we get these proteins here called TET1 and polycomb. Now, polycomb is extremely specific to certain areas of the genome, and it binds particularly to the enhancer of vasopressin. When it binds to the enhancer of vasopressin, it recruits MECP2. Now, MECP2, interestingly, only becomes activated and expressed more during the postnatal period from days 1 to 10. After that, it decreases. So it's during this period that stress deactivates MECP2, and so you don't get the binding and you don't get the methylation here. And later in adulthood, this methylation is activated, is consolidated with depressive chromatin and DNMTs and HDACs. I won't bore you with the thousands of chip experiments. But what happens is you get the recruitment of this various chromatin through DNMTs and HDACs, and this leads to reduced vasopressin, reduced HPA axis, and reduced stress response. And understanding all of this is crucially important. So if you know the different players in this pathway, we can target them. So for example, I've shown if you add DZNEP2, which inhibits polycomb binding, you can actually regulate vasopressin a long time later. You can also inhibit HDACs through valproic acid, and I've also shown this regulates vasopressin. You can inhibit DNMTs to bisphenol A, and you can also inhibit DNA methylation through 5-azacitidine. All these factors can be modulated with drugs to actually change long-term regulation of vasopressin. Of course, these aren't so specific to the vasopressin, but all of these obviously regulate. So coming on from this then, it was clear that there's something involved with the pups, with the mother, that was causing this long-term stress activity. So I then looked at a rat model and developed a rat model with Ben and nephew, and we showed that measures of maternal care and depression by a social stress, that's what we did, we socially stressed the mother rat with the pups. This reduced levels of maternal care, and this caused the infants to develop a higher stress response. The levels of maternal care we found were levels of maternal licking and grooming particularly. And these were modulated through changes in endocrine and immune changes, and these were regulated through epigenetic changes at particular endocrine and immune genes. So then went to some human studies. So I've been working with some excellent clinical psychologists, and we looked whether maternal care also relates to some of the human maternal care in the rat to the mouse. So I've been involved in this Liverpool study, which is funded through the MRC for 3 times, and I've been looking at maternal depression and various other stresses, prenatal and postnatal, and their impact on childhood behavior. This is in the Liverpool region here on in the rural. So we looked at the glucocorticoid receptor, which was a gene we found was regulated in the rat, and a few other studies have looked at depression as well in the glucocorticoid receptor. We looked at that, and we tested if you get similar effects with maternal care. We looked at maternal stroking. So we found that the rats that licked the pups more, the level of maternal care with the licking and grooming actually had a dampening effect on the stress, and also regulated the glucocorticoid receptor methylation, as I showed. So looking at this in humans, we had evidence within the mothers who reported their levels of stroking. Now, those mothers reported their levels of stroking show that they actually dampen the effect of stress. So in the dashed lines here, what you've got is low levels of maternal stroking. And in response to increased on the y-axis, in response to increased depression, these children on the x-axis showed increased levels of stress behavior, this IBQ, the stress. But those mothers that stroked, even following high levels of depression, these infants did not show that level of stress reactivity. So it's very similar to what you find in the rat. And then when looking in the CpG methylation, the same gene with the methylation, we see that on the right-hand side, the dark line, we found a reduction in glucocorticoid receptor methylation following higher levels of stroking by the mothers. So you find very, very similar things going on. One possibility for this could be the idea about this receptors we have in the skin, the C tactile neurons. So these are in our skin and these respond to touch. And I work a lot actually with Frances McGlowan and Suzanne Walker at Liverpool John Moores, who are experts in this. The other thing we've been looking at then is from the human study. Now, from the human study, we were thinking about the PAR hypothesis. Oh, sorry, I'm trying to moving that down. So we're thinking about the PAR, so the predictive adaptive response hypothesis. So this is this mismatch theory. So the idea is that early in life, as I've said on the very first slide, that you adapt to your later life environment. So postnatal life, you'll adapt to the later life. Now, one possibility is, we thought, is if we have a prenatal adaptation to the postnatal. This has never been tested before and ever been shown before. So we tested that in the rural study that we had the DNA for. And on the same study we had with the stroking, we found that those infants with the low prenatal depression in the dark line here on the left-hand side, in response to high levels of maternal postnatal depression, so postnatal stress, actually responded with high levels of stress. And it also responded with high levels of glucocorticoid receptor methylation that correlated together. Now, those infants with high levels of prenatal depression did not respond to the increased levels of postnatal depression. So in the gray line, so the ones in the gray infants, they actually had the prenatal depression, actually protected themselves from the postnatal depression. They actually adapted to expect higher levels. So it's not what you might have thought. It's like prenatal high depression and postnatal high depression give high levels of stress in the infants. It's completely the opposite. And this again follows the idea about adaptation. So from the back of that, then I designed an animal model that we just finished actually. So my PhD student Laura here we designed an animal model for prenatal and postnatal stress. So we have all these different conditions here with prenatal and postnatal control, prenatal stress only, and no postnatal stress, no postnatal stress, and no prenatal stress and only postnatal stress, and pre and postnatal stress. And so the ideas was that those animals on the third one down, those without the prenatal stress but just the postnatal stress, will show the highest levels of stress response as adults, and also the highest levels of glucocorticoid receptor methylation. And that's what we're doing at the moment. So we have some data to support that at the moment, but I'll let Laura show that. But the important thing is, I want to say, is there's no way I could have convinced my PhD student Laura or Joe Neal I work with to do this if we didn't have the evidence we found in the actual human studies. So the human study was perfectly there to be able to support the design of the animal study. And within the animal study, of course, we can look in the brains, which is what we're doing, and give a far more conclusive understanding of the actual mechanisms under play here. So the other thing I've been looking at is the intergenerational inheritance then. So the idea is that if you've got stress during early life, can it be passed on to further generations? So I'm calling it intergenerational inheritance. And the idea is that we'll get changes in the behavior. So what you found is that looking at chronic stress, those pups exposed to chronic stress at the mother's level, so this chronic stress is a resident intruder, which is kept away from the mother, so there's no actually any contact, it's just a psychological stress with a male in the next cage. These pups then will then take that experience from the mother, and then when they become a mother, that experience will be passed on. And so we've looked at over 3 generations. We've looked for epigenetic changes, behavior, endocrine, immune, and also lactational as well. So to cut a long story short, after a lot of research has gone into that, what you found quite interestingly is rather than having this low level of depression in the first generation and it being lost, it's actually snowballed. So if you look at the first graph here, the maternal care, during a social stressor, that goes down. and then the animal recuperates. On day 16, the care is normal. In the F1 generation, day 2, those animals in the F1 that were subjected to the stress in the mother, they showed reduced levels of maternal care only in day 2, but then they actually recovered, and day 9 and day 16 were okay. Now, in the F2 animals that were subjected to low levels of maternal care in the day 1 mothers, so the grandchildren of the original FO, these showed decreased maternal care, massively decreased maternal care through all days. So it's not something that's lost, it's something that's become adapted. Now, interestingly, you also get other changes such as maternal restlessness, which is a sign for maternal depression, and we get a reduction in maternal aggression, which is a reduced level of maternal care as well. Interestingly, intake originally was affected in the FO, but this is completely normalized in the F2. So the F2 animals have actually adapted with reduced levels of maternal care. They've got normalized milk, and many other things become normalized except the levels of maternal care. Interestingly, corticosterone in the F2 animals is massively increased, and this associates with very strong acute stresses, whereas in the F2, associated with chronic stresses, we get a decrease in corticosterone. We get this hypocorticosterone excretion, which is strongly associated in many recent human studies, also with chronic stress as well. Interestingly, we found a number of genes dysregulated and a number of epigenetic changes, such as in the GR, which are no longer evident in the F2. So something else is adapted in the F2. We're also doing a load of other things, such as RNA-seq, to try and understand what are the actual differences between these FO to F1 and to F2 to see where this adaptation changes are actually occurring. So it sees many of the other endocrine changes seem relatively okay, but obviously there's something else happening here. Interestingly, when we know some of the mechanisms underlying this, and we know within the F2, I didn't show you this, but we showed a whole host of other endocrine changes. Some of these were reductions in oxytocin, which is important in maternal care, and vasopressin, again, which I showed you is important in stress. So we know these are important, and what we did is that if we give intranasal vasopressin and oxytocin to the F1 mother, we completely normalize the reduced levels of maternal care and maternal depression-like phenotype in the F2. So you can actually stop that by targeting that neuroendocrine pathway. And this is really interesting. And we also found changes in the immune sensitivity as well. So you actually got an increase in interferon gamma following oxytocin, suggesting that an increase in immune response is also important in reversing these maternal care and maternal depression phenotype. So going back from this then, from the animal studies, we've gone back to the real study at the moment. And what we're doing is we're collecting data from how the mothers who were looked at with depression, how their perceived levels of maternal care. So the grandmother's maternal care. And what we're doing now is looking at 3 generations across the human study and looking at oxytocin and GR interactions to understand if they got this intergenerational change that we found in the animal model there. And a couple of recent evidence is showing, particularly last year, that mother's exposure to early life stress increases the child's risk for behavioral problems. So this could be a major mechanism towards child stress and child mental health. And this data also supports a number of other studies. So just this morning, in fact, I'm speaking with someone from a colleague I work with in University of Oxford, working on as well. We've had a big sort of family trial looking at family-centered treatments to tackling child mental health. So it's not simply looking at the child itself, but to actually involve the mother as well. And it's treatments really aimed at the mother to try to see if to reverse stress-related changes in the infant. And I've got DNA from that study in the freezer at the moment. And we're looking at the same genes I showed you and in the for methylation in the animal model I showed you. So it's really the animal model leading the way towards human studies here. Now, another thing we've been looking at is this level of social stress and immune changes as well, which has been on the back of many of these studies. So the maternal depression study, we have another social stress study as well, where we swap these mothers around within different communities, and that's been a real social stressor in the animals. Really psychological social stressor. And also following the odds of vasopressin oxytocin intranasal, again, many of these studies are finding the same candidates coming out with things like TNF alpha, IL-6, CRP, and ICAM. And I've also worked with a study that's recently been funded by the DFG in Germany to look at immune activation. So many of these factors are increased and altered following stress, but many of these factors also protect. So with immune activation, you've shown that if you immune activate early in life of a pup, you actually protect it from stress later on in life. So what we've been funded for at the moment is to give immune activation in pregnant mothers, rats, and see how that affects the stressor. And the idea is that immune activation, this jolt of the immune system, actually is a protection against the stress. This will be working in a different way, we think, to the chronic levels of immune, of like age-related inflammation.
3 minutes.
Yeah, thank you. So level of social stress we're looking at in the human studies has been a study I'm looking at the moment now, which is the Latina mothers in the US, particularly following Trump's election. So what we've been looking at is we've been looking at this level of discrimination. So these mothers reported discrimination, and we've also got levels of prenatal depression and various other measures, and levels of postnatal depression. So what you see is in response to discrimination stress, like a social stressor, we see the same changes in glucocorticoid receptor like we see in the animal studies here in the top bit. Now, in the bottom bit, which I published, we see that those mothers with higher levels of TNF alpha and higher levels of FOXP3, suggesting a higher immune activation, these are, following the methylation, these are more resilient to the effects of discrimination in inducing postnatal depression. So those mothers with the higher responding immune systems are actually more protected against social stresses. Again, what we find in the animals. Currently, I've been funded to look in many of these experiments I've shown you in the Ribble study, to look in India as well, in Bangalore. And many of these hypotheses I've shown you now are also being tested in Bangalore at the moment, in fact. Now, the final thing I want to finish off with is the G by E, the gene environment. So the idea is that everyone may be exposed to an environment stressor, but not everyone develops a depression outcome. And it's the mixture between G by E. So you may have a genotype, and following early life stress, this genotype will determine the methylation in the gene, and this will determine how the gene is regulated. So it's not just the early environmental stressor, but it's the genotype you have. So looking in dementia, and I've done quite a few studies in depression on this, but to bring this towards dementia, for example, in interleukin 6, there's a polymorphism which is well associated with Alzheimer's disease. And this polymorphism changes the CPG site. So the G is the CPG site, whereas the C doesn't contain a CPG site. Now, those people with the G polymorphism have high levels of methylation at CPG2. You can see here, and those with the C polymorphism don't have any methylation. And methylation of the neighboring CPGs are also affected. Now, these people with the CBG phenotype have the G there. They show lower levels of protein, and they also show lower levels of methylation within the brains following Alzheimer's disease. So you have this evidence of a CG interaction within the brain of Alzheimer's. And another thing important in developing these animal models are, for example, I looked at BDNF for another CG G by E interaction, and this did not occur. So we did not find any G by E interaction in these brains of Alzheimer's, but we had relatively few individuals for that. So what we're working at the moment is on a humanized BDNF G by E. So we've got a humanized rat, and what we've done at the moment is we've given this rat stressors, and we've given these rats exercise, and we've looked at cognition. So cognition during the ages of these animals, just to look at methylation. So this DNA is also sat in the freezer. So as soon as I can get in after the lockdown, I can look at that as well. And this will allow me to understand how the methylation is working at this polymorphism, to understand better the GBE that I can target back in those human studies that I wasn't able to find. So just the last slide really is the animal models for G by E. So the idea, what we're looking at the moment, is to look at numerous different mouse lines with different genotypes, and looking how these are affected by similar early life stresses. And this is an idea I've got at the moment in submission to try and understand the genome-wide approach to understanding G by E. And so hopefully I've managed to convince you the importance of animal models in determining studies in humans, and vice versa as well. So I'd like to finish up with thanking a lot of my collaborators from South Carolina with the Latina study, Liverpool with the Ribble study, and King's College as well with the Ribble study, Bangalore I work with within the Bangalore Child Development, Reading, Manchester, Max Planck, and University of Oxford. And for just 10 seconds, if anyone's teaching genetics at the moment, I've got a new book out, which is called Conversational Genetics. And so it's basically examples behind genetic disorders. So I thought I'd give a plug in there if anyone wants to think about buying that for Christmas present. Any questions? I went through that quite quickly, so I hope you are able to follow. You're muted, Sonia.
Someone muted me? Okay, so we have a few questions for you. So the first one, PVN neurons have important roles in the modulation of nociceptive information. Do you know if maternal social stress affects nociception modulation? PEN, basically.
Yeah, that's really important. That is, and I've got me because I was working on something. I'm working on something like this at the very moment. And I was speaking to someone about this, in fact. So yes, nociception is highly related to stress and depression as well. There's not enough research gone into this, and this could be really important as well. We didn't do nociception in the animal model. Annoyingly, we were going to look at this at one point, but we weren't able to get that through the ethics. But that would be a fantastic thing to do, and I think that's a very, very important area to be looking at.
Okay, so there is more questions here for you. So how does the change in F0, the first generation, I suppose, are passed on to the next generation?
Yeah, so I couldn't go into that as much as I wanted to. This was my last
PhD
student's project. So yeah, 1 second. Okay, well, give me like 10 seconds, I can go through it. So the one thing is through the levels of maternal care. So the levels of maternal care were basically changed with that. We could see that. But interestingly, what I couldn't show you is that even within the milk, we found different levels of hormones within the milk of those mothers that had been stressed as well. And so the main idea we're looking at is it's passing through the mother. So the mother shows depressed phenotype, and that depressed phenotype is obviously impacts the offspring. The offspring don't get the levels of maternal care they would normally get. The reduced levels of licking and grooming, the reduced levels of the increased levels of stress that the offspring pick up. And it's that's taken on to the next environment and then to the next generation.
Okay, one more question then from Rodrigo. Are you currently working or seeking to work soon with computer or algorithms for predicting the intergeneration transmission of stress, or your approach is mainly statistic? You're gonna model or carry on using stats, basically?
If he's offering to do some mathematical modeling for me, then I'll say very happy.
Okay, then please get in touch with Rodrigo. Rodrigo, get in touch with Chris. Right then, so let's move on to the next question, the next talk, because we are run out of time, isn't it?
Don't worry.
Okay, okay, let me introduce then. So the next question, the next talk, is from Dr. Monica Lopez Hidalgo. She has obtained her bachelor degree in biomedicine and her PhD in biomedical sciences at the National Autonomous University of Mexico. After that, she completed her postdoctoral research at Max Planck Institute for Neuroscience. Well, maybe you met Chris there, didn't you? And then she's now currently a professor at the National University of Mexico, where she leads the undergraduate program of neuroscience. Her research focuses on the role of astrocytes in cognitive function and sensory processing. And her research is internationally recognized. And in 2017, she was awarded the L'Oreal UNESCO Fellowship, I suppose, for Human and Science. So, Monica, welcome, and please, it's with you. Thank you.
Thank you very much. Really a pleasure to be here and share with all of you. So, last session, we could see some of your views. You were very kind to share with us some of your images from your campus. So today I decide to show you some of ours. And I assume the students are going to feel like nostalgic and wanted to come back here. So we live in Querétaro. This is where the institution is. And we have beautiful weather. It's windy. Yes, it's cold sometimes. And sometimes it's very warm. And it's a semi-desertic area. So we have this kind of nature. And it's actually really nice. So we also have like the visits of butterflies like these ones. And usually around these times. And it's really spectacular to see it. But actually what I like the most in the campus are the sunsets. The sunsets are really spectacular. So I'm going to share with you some of them. This has been taken with just regular cameras from your cell phone, no filters. So you can see there's so many colors. There's so many beautiful stuff there. It's just really amazing. So this is our campus. This is NS. And it's so beautiful that Jorge Luis Borges wrote in the book El Aleph, vi la noche y el día contemporáneo, vi un poniente en Querétaro que parecía reflejar el color de una rosa en Bengala. I saw the contemporary night and day. I saw a sunset in Querétaro that seemed to reflect the color of rose in Bengal. And it's really, really nice. So if it's possible and then you can come here once this is over, then we're gonna be really happy, and then you can also watch these beautiful and spectacular sunsets. Okay, so let's get this started. So I'm gonna talk about aging in this ring, but then I need to talk a little bit before. So why are we starting working with aging in this ring? So actually, my lab is focusing on neuroglia interactions, and in particular, I'm interested in astrocytes. So right now, my lab members are working in these three different areas: the role of astrocytes in sensory processing, in cognitive functions, and also in aging. We all know that astrocytes are really important in regulating homeostasis, which is really, really huge and really important for just anything that the neurons wants to do. So it's ion balance, metabolism, neurotransmitter levels. But more than these homeostatic roles, we know that astrocytes are doing more interesting stuff. So astrocytes establish a bidirectional communication with neurons. That means that they are morphological attached, close to the presynaptic and postsynaptic neurons. And we have this kind of figures representing the physiological interactions of astrocytes and the synapses. We have receptors on the processes, and we have receptors also on the pre and postsynaptic neurons that can sense what the astrocytes are sending. So there's a lot of astrocytes covering gray matter, but this is really not a good representation of what astrocytes are really doing to communicate with neurons. This is maybe a better way to show it. So there's a plethora of membrane receptors. That means calcium channels, metabotropic receptor, iotropic receptors. Astrocytes are able to respond to neural activity due to increases in calcium activity. And then sometimes it can occur due to a calcium elevation, a release of gliotransmitters that can potentiate and change the communication between these neurons, and also have an important role regulating blood vessels. So this is really a complicated story. And so astrocytes are really complicated and really different, depending on where they are and the age of the animal, the species, etc. So besides these roles that has been really well described, it has been like in the last 15 years, 20 years maybe, talking about synaptic transmission and synaptic plasticity. So almost a decade ago, I started working with synaptic plasticity. And so I was wondering what is the role of astrocytes in one famous type of synaptic plasticity, that is LTP. So we were analyzing what is the role of a molecule that is released by astrocyte, that is this ring. And we use hippocampus as a model. We use slices, and we were stimulating the Schaffer collaterals and recordings on CA1 pyramidal neurons. And we were having just a regular protocol for analyzing synaptic transmission and plasticity. And we were wondering what astrocytes are doing in this kind of synaptic plasticity. So then I have here the basal responses. And then we have a high frequency stimulation. We have a LTP, that is an increase of the responses that remains for a long time. And then we use a drug. At that moment, there was not like so specific agents to block astrocytes. So we were using something that it was good at that moment. I will not recommend it right now, but it was a fluorocetate that temporarily blocked activity of astrocytes. And what we observed is the LTP was not able to establish. So then we thought, what astrocytes are releasing? So then if we blocked the astrocytes, the LTP is not observed. So then we thought, we knew, because there's many information about that, the role of NMDA receptors in this LTP. And so we have two candidates for this. So maybe astrocytes are releasing glutamate, that it's agonist of NMDA receptors, or it's maybe releasing discerine, that is a co-agonist of NMDA receptors. So we choose discerine, and then what we observed is that when we apply discerine in the bath, we were able to fully recover the establishment of LTP. So that means that discerine released by astrocytes modulates NMDA receptors and it's able to maintain synaptic plasticity. So then we wonder if this has a correlation with memory, with learning and memory, because we know LTP is the cellular basis of these processes. So then if we were able to restore discerine, what happened if we try with learning and memory? So just to make the talk shorter, I'm just gonna show you just one result. So we use inhibitory avoidance task to measure a long-term memory, and then this is what we observe. So this is the retention latency. This is a measurement of long-term memory. This is the control condition. This is the ones that receive the inhibitor of astrocytes, fluorocetate. We have an amnesia. The rats didn't remember. If we administrate just discerine by itself, it doesn't improve. But if we administrate discerine plus fluorocetate, then we were able to recover the memory. So then that was like 8 years ago. And then I was working with another projects, but then so we knew that astrocytes were involved in many stuff, including cognitive functions. But then most of the information was obtained using young animals. And then what about aging? And we know that cognitive start to decline with aging. And we were wondering if discerine were also involved in this cognitive decline associated with age. Okay, so then this is how we start to thinking about aging and discerine and astrocytes. So then aging is associated with many morphological changes and functional changes, many more. And there's a lot of theories about what is happening, and then it's a multifactorial event. So we know that it's affected synaptic plasticity, attention, cognitive flexibility, memory, and many more. So we saw that there was a common path, and it was like all of them are NMDA receptor dependent processes. So then we wonder if all of them were involved in NMDA receptor. And in fact, there's if you see the development of the cognitive performance, like I'm showing here, this is in human years. So then it has been proposed that this decline of the cognitive performance is related with an hypofunction of these NMDA receptors. So then we thought, like, what if this NMDA receptor is affected because there's not enough discerine? And in fact, it was published in 2009 that with age, there is a less synthesis of discerine. So then that was a good hypothesis. What if discerine is in part responsible of these changes? Because we showed in young animals, so we wanted to try with all animals. Okay, so then we start testing cognitive flexibility, and we use rats to do that. So let me show you briefly the task. So this is a rat. It's in a box, and then the the rat learns how to press ipsilateral lever related with a light. So then the rat is there, and then there's one, you can turn on the light, the ipsilateral or the contralateral, and then the rat must press the ipsilateral lever in response to the light. And if the rats do well, then it's going to receive a reinforcement. So then this is a cue, this is the light, and then this is the ipsilateral pressure, and then it's receiving a reward. If the animal press the contralateral lever, then it's not receiving any reward, and the animals are food deprived, so they really want to press the good lever. So then they have an 85% of performance. They are really good doing this. It's a really simple task, but then one day we change the rule. So then we present the light, the rat comes, press the lever, and then there's no reward, and no reward, and no reward. And what happened is the rats just like is desperate, because usually that works, and then that day doesn't work. So then it's trying to press and press and press and press, and we count these perseverative errors as the times that the animal is still trying to use the same strategy has used, instead of trying to look for the other lever that now is giving the reward. So then let me show you here and a video. So this is during the cognitive flexibility test. So here's the rat. So then it's not pressing, it's just desperate. It's light, nothing is happening. It's not receiving a reward. It's pressing and pressing and pressing and pressing and pressing. And they're really desperate. It's like they don't understand why it's not working. I mean, for 23 days, it works really good. They press the ipsilateral lever and they receive the reward. This time, no. Okay, so then we decide to, I'm gonna go back a little bit here. So then we decide to test this hypothesis. If this serum was involved in the cognitive flexibility, and we wanted to see the cognitive flexibility that is decreased with age. So then we choose two different ages that is related with human ages. It's around 60 years old in humans and 45, which is real, because as you can see here, we start to decline our cognitive functions around 35 years. So then this means this is 18 months in rats, and this is 24 months old rats. Okay, so then I have here, let me change my laser pointer. Okay, so this is the histogram. We have here how young behaves, middle age, that means 18 months, and 24 months age rats. As you can see, there is a dephase to the right, which means the rats are really having more perseverative errors in general. As you can see here, compared with the young, the middle, and the age have a lot of more perseverative errors. That means that they have less cognitive flexibility. So then we wonder if this serine was involved in the aging-associated cognitive flexibility. So then if this is true, then we supplemented this serine in the drinking water 2 months before they were evaluated. So then when we see the results, this is the young again, and this is the rats that were treated with this serine. And there was no difference among all of these worked, which actually we were really surprised, because I mean, we thought like we will have an effect, but it's not like we will recover all the function. So then we said that aging-associated cognitive flexibility decline is prevented by this serine, but then we were wondering what are the mechanisms mediating this serine cognitive effects. So then we went to the a general technique, a broad technique analyzing the brain, because we wonder if there was many regions affected, because we were given this serine into drinking water, so potentially it can act in any part of the brain. So then we use fMRI. We use fMRI as a tool and as a technique to measure the changes due to this serine treatment. So then we use resting state, when we use an NBS statistic model to evaluate different changes in different cortical areas. And then there was 3 that was significant different, and it's frontal, cingulate, and retrosplenic. So then we have here the functional connectivity, that is what we obtain with this resting state from young in compared with middle-aged and aged animals. As you can see, there is a decrease in the functional connectivity of this frontal and cingulate communication. And when we supplement this serine, then we recover this decrease in functional connectivity between these two areas. So then we went to check the other connection, and then we observed the same. It was even a bigger decrease due to age, and we were able with this serine to restore again this cognitive functional activity. Okay, so then this serine improves functional brain connectivity in aged rats. But what about now an histological point of view? So we know that these frontal cortex are affected by age, and one of the affections that you can see, it's at the histological level, is there is a decrease in the branching levels and in the dendritic trees, but also in the spines. And it has been very well documented that it's associated with age. So then we wonder if the treatment of the supplementation of this serine could improve this loss of dendritic and spines. So then it almost didn't change the dendrites, but it actually modified a lot the spines. So then here we have a classification of mature and immature spines, and then we have an increase of the number of spines. I'm sorry. Yeah, and we have an increase in the number of spines, mature and immature, in the middle-aged rats, but also in the aged rats. That means that this really increased the number of spines in aged rats. Okay, so we have very limited time, so I'm just gonna summarize some other results that goes with this slide. But if you want to talk about this later, I'm really happy to do it. So is this serine able to restore other cognitive functions? Is this only applies to cognitive flexibility, or is it really that this serine is a cognitive enhancer to all functions? And so we test attention with the rats, and we didn't see any effect. So it's really specific for the task, at least for this. So does this serine works with a lower dose, because we are using 300 milligrams per kilogram of weight, and then we were wondering if maybe a lower dose, so we use 30 milligrams and no effect. So then we wonder if is this serine able to improve cognitive flexibility in young animals. And actually this was proposed for my students because they are young, and then they were always excited about the results, and they always joke about taking this serine before the exams and everything. So we decided to do the group, and unfortunately no effect. So it seems like it's really dependent of the type of function you are analyzing, depends of the doses, and depend of the age of the rats. So then our conclusion is that aging associated cognitive flexibility declines prevented by this serine, and is acting through frontal cortex, changing functional connections and histological changes into the neurons. So finally, I would like to thank all my lab members. I have only women, I don't know why. And so all of them, some of them are undergraduate students like Frida and Barbara, and so they were doing most of the experiments. And of course, my collaborators, that they are always assisting me in many ways. And of course, I would like to thank the funding, because without the funding, we would not be able to work. And thank you very much to all of you.
Well, thank you very much, Monica. It seems that I'm frozen there, but can you hear me?
Yeah. No, I'm not frozen.
Oh, okay. I'm better. My computer probably was. Oh, thank you very much for the exciting talk. And we have some questions for you, but I will first start a question for myself, because I'm pleasantly surprised that you are interested in synaptic plasticity. I work on synaptic plasticity, and we are even interested in the same synapses, Schaffer collateral CA3 to CA1. So what is the area of the hippocampus that I work on? So I have a question for you. So did you record for the hippocampus of these rats after you did the training?
No, we It's a different set of experiments. We did the recording in slices, and then the in vivo, it was apart. So no, yeah, that will be a great experiment. You know what we want to do now to kind of analyze this kind of questions is to do supplementation of viscerin while the rats are under the fMRI, under the resonance, so that we can see the effect of acute viscerin into the whole brain. And then we can also we are doing now longitudinal analysis to see how this changed the brain after. So we are now doing the longitudinal analysis. That will be a great experiment, but unfortunately, no.
Yeah, no, it would be great, because we work in spatial memory, and we record, do all the experiments, learn, and then reversal learning. And then when we finish the experiment on the same day, we kill the animal, and then we record for the same animal that we did the behavior, because then you can have a correlation between both, what is going on that synapse and what you are seeing on the behavior. So I know that there are plenty of questions there, but I have one more question for you. So did you try any other spatial hippocampal mediate tests?
No. We were thinking in using water maze, but at the end we decide inhibitory avoidance task, and no. And I moved now to the frontal cortex. I'm not working anymore so far yet with the hippocampus, but I would like to come back, because also hippocampus has a really important role modulating frontal cortex, and it's affected particularly with age. So I'm thinking going back to the hippocampus and then check some other stuff.
Mainly because the lateral and the medial frontal cortex projects to the hippocampus. All the information is processed in the hippocampus, integrated itself between them, but also the information comes to the hippocampus to be integrated, isn't it, into the hippocampus? Yeah. Because I think you have an excellent model, in my opinion, to test the reversal learning. Then you could probably test really the cognitive flexibility, is the ability to learn a task and then how to reverse it. And for example, Moore's water maze, I'm not entirely sure, but Barnes maze, I know that Barnes maze with rats it doesn't work. We work with mice because we use a lot of genetic modified, but that would be fantastic for your model to really test the function, in my opinion, the discern importance in the cognitive flexibility. But we can talk about that in another time, because there are other questions for you there that I'm going to pass to you now. So Raul Gerard Paredes Guerreiro asks you, is there a sex difference? Is astrocyte expression in the brain? In the astrocyte expression in the brain, are the effects of discern similar in males and females?
This is such a great question, and I don't have an answer. And because I have only female members in my lab, we always say, like, we should do a comparative work and analyzing females. And I have done it, but yeah, I'm gonna do it, because that's a great question, and we need to start doing biomedical science, putting females into the groups and then analyzing it. I'm pretty sure it's gonna be quite different.
Yes. So yeah, no, I agree. I totally agree as well. Probably everybody does. So there's a question for Neda. As aging significantly causes a decrease in D-serine content, can we reverse the whole of aging by increasing D-serine levels?
I'm sorry, could you say again?
So as we have decrease in D-serine, with aging, so the levels decline. So can we reverse the whole of aging by increasing D-serine level? I think you answered that already.
Yeah, I mean, that's the general idea that we try to prove. Like, if we increase D-serine level, then that will change. But it's not. It's really specific. It's not like it's reversing aging. And but something that it's also interesting is that there's NMDA receptors in the tissue, in the peripheral tissue. So in the bones, in the muscles, in the gut, in kidney. So there's NMDA receptors everywhere. So we are now writing a perspective about really answering this question, but not only in the brain, like in the general. So then NMDA receptors are also important for the other tissues to work properly. So then maybe it's a hypofunction of NMDA receptor. Maybe it's a decrease of discern in this peripheral tissue. And that's why also when people age, or when the animals age, it's just a change not only in the brain, it's a general change. So then we are working right now in these questions, and in fact we have a collaboration with one of our colleagues at NS. So we are going to analyze the bones, because the bones are really dependent of NMDA receptor activity, and we have the bones of those animals that we analyze, and then we are now going to measure how they resist to be broken, and if this ring changed this flexibility and strength. So we were about to measure just when we start with this COVID, and this is stopped, but I think we are rescheduling for next week. So hopefully in 2 weeks I will have the results. If really this ring is reversing aging effects, not only in the brain, also in the body.
Oh, that's very good. We are looking forward to hear that data. So one last question for Lloyd Orton. I think you met last week when he gave it, or last time when he gave a talk. So very interesting and clear talk. We have unpublished data in age ferret showing that at 5 years they performed near ceiling on an auditory cued task similar to this. But when we measured their hearing thresholds, they have severe to profound hearing loss. Do you think there is any influence of sensory disturbance on your age behavioral data?
This is a really great question. So yeah. I mean, at the beginning, we were deciding what will be the task for testing cognitive functions, so like the lights or sounds, and we know that the hearing and the sight, it's affected with age. So we thought like if we put like a more complicated task with discrimination of different hertz, that will be a problem, because maybe with aging you have problems to detect the stimuli, and then your performance is not going to be good. So we choose a task that is really, really simple, and then it's just a light that you have to just see, and it's in front of the rat. So we don't think the rat is seen well, is able to move and everything. And in fact, we have the reaction time of the rats. In the reaction times, yes, it decreased, but it's probably affected by the locomotor and all these motor parts. But it takes a little bit longer for the aged rats to get to the efficiency, the 85%. It took like 3-4 days more. So it's like the curve is a little bit less pronounced. But at the end, they reached exactly the same level of performance. So in 23 days, rats all age reached exactly the same level. So I don't think there is a sensory problem in detecting in my animals. However, I have to say also that astrocytes process sensory information, and that's other part of my my current work. And in fact, I was working with ferrets before in visual cortex, and we know that the organization of astrocytes change in 2-year-old ferrets. So thinking in the role of astrocytes in processing and how this could affect the brain in aged animals, of course, this must be totally affected. That will be like a different role of astrocytes, maybe not only with discernes, maybe more pathways.
Of course, that would be interesting to have some models where we could, avert it, isn't it, to activate the in cells, the release of that, but discern, and so many things to do. So if we don't have any more questions for Monica, So I think we are on time as well. So I would like to thank you, Monica and Chris, for the excellent talks. And it was very nice to talk to you all. And hopefully we can discuss some research projects in the future.
Sure.
I think it would be very good. I'm really interested in, in your work, and mainly because we have similar interests. Some glutamatergic synapses, and microglia. We are interested in microglia, and it'll be very interesting.
Sounds great.
Do you have anything to say, Rita? So good.
It's all good. Thank you to speakers, and we'll see you next week for the last of this series of neuroscience webinars.
Okay, well, thank you very much for having me. Thank you.